Welcome to our first podcast in Chapter 4. We're beginning the third week of development, and we're going to go through the process in this podcast of gastrulation, forming the primary germ layers that we're going to lay down. That's going to make up our embryo. It's going to make up all of the structures that we know in the adult. And you can see in these some illustrations, and on the far right, you can see basically the process of gastrulation converts a single double-layered disc, our embryonic disc, into, at the bottom, a triple-layered embryonic disc. We're going to have our ectoderm on the surface, endoderm on the bottom, and mesoderm will be scattered throughout the middle. Now, these processes are going to be absolutely critical during this third week. We're going to begin the process of gastrulation in our embryonic disc, and that's what we're going to focus on. We're going to leave the rest of the developing placenta behind at this point and focus on the disc. In the posterior zone of this disc, on the very surface of our epiblast, we're going to see a line drawn in the sand. This is the primitive disc, and this is going to be the beginnings of gastrulation, the formation of these primary germ layers. Now, in addition to that, we will go on in subsequent podcasts to talk about the formation of the notochord, neurulation, the formation of the body cavity, all of these critical events that are occurring during this third week of development. So here we're going to focus on gastrulation. And gastrulation is the process where we take this bilaminar embryonic disc that consists of an epiblast and a hypoblast, and we're going to convert it into our trilaminar embryonic disc that's composed of our endoderm, ectoderm, and mesoderm. Now, this also marks the beginnings of the process of morphogenesis, where we're really developing body form. Because as soon as we make a point and a line in our disc, this primitive streak, we begin to determine body axis, which is going to have a critical impact on body form. And so as soon as we see the primitive streak, this is going to be the time in which we will now refer to the embryo as a gastrula and no longer just as a blastula stage embryo. So as we look at the primitive streak, we're going to focus in the posterior portion. We call this the posterior marginal zone, or PMZ. We're going to see a depression first. And from this depression, we are going to see a line move towards this posterior margin. And we're going to see a video that's showing this, in which the cells that are beside this groove are going to become taller in nature. We're going to have a primitive groove. We're going to have a primitive pit. We're going to have all of these landmarks that denote this very critical event that's beginning the process of gastrulation in the posterior portion of our embryonic disc. So here we can see our embryo disc, outlining it as such. Anterior is the top, posterior marginal zone is here. And this is our primitive streak. The primitive node that you can see here at 7, this is the beginnings of this line. And the primitive streak runs toward the posterior margin, the posterior boundary. The white line that you see in the middle is going to be the primitive groove, and it's basically leaving, you can see these darker edges on the side. These are raised areas where the cells are becoming taller on either side of this depression. So it's almost going to look like a trench that has been drugged through the epiblast of the embryo beginning at the surface. I think about it almost like when you're at the beach and you push your finger into the sand at the beach. You have a nice smooth surface. Your finger goes in here. That would be synonymous with the primitive pit. And then as you draw your line backward, that's going to leave that groove that's going to pile the sand up on either side, and that's going to be fairly reminiscent of what we see with our primitive streak that's happening again in the posterior marginal zone. And you can see we've got this anterior portion of the embryo that's untouched at this point. Actually, all of our embryo is going to develop here. The primitive streak over time is going to regress and completely go away. This is just an inductive center. It's a place where we're going to begin to form our germ layer cells and distinguish them from the epiblast cells that we have on the surface. Remember, everything that we are and are going to become is derived from the epiblast. The hypoblast cells are almost like placeholders lining our umbilical vesicle, that secondary yolk sac, and those cells are going to be replaced by migrating cells from this epiblast that are going to pour through this primitive streak, migrate down, and replace those epiblast cells, and that's how we get our endoderm. So in our illustrations from our text, here we're looking at a cut view of the amnion, looking down on our embryonic disc. Here you can see our primitive streak in this posterior marginal zone. Here we have our umbilical vesicle, that secondary yolk sac. And these arrows are trying to show you that these epiblast cells are moving almost like a conveyor. They're moving toward the primitive streak, and some of these cells are going down into the primitive groove and underneath. Now, at this level, if we were to take this dotted line, if we were to make a transverse cut through our embryonic disc at the level of the primitive streak and look at that cut edge, what we would see comes out in D. Now, developmental biology is very much about three-dimensional thinking. So here's our amniotic cavity. Here we have our umbilical vesicle. Here we have our primitive streak that we've cut through. This is our epiblast in blue. Our hypoblastic cells are here in yellow, and I, again, don't like this term endoderm yet. We haven't produced it yet. But you see these arrows, they're moving in from the groove and underneath. This is how the migrating cells of the epiblast are going to get down into this region. The first cells through are going to replace the hypoblast to become the endoderm, and the next cells are going to stay in the middle, and those are going to become our mesodermal cells. So the primitive streaks really is a critical concept. I heard in graduate school that gastrulation is the most important event of your lifetime, because if you don't gastrulate, you don't develop. Well, relative to gastrulation, the primitive streak is a critical concept, and in 2002, there was a President's Council on Bioethics, and this was a finding about human cloning and human dignity. And so this finding was that the primitive streak is going to be the signification of the creation of a unique potential human being. And I guess that potential human being suggests that once you see the primitive streak, you know something's happening. Well, but I think you can already appreciate from the time of conception, when you have a fertilized egg, things are happening. Even if we can't histologically see them under the microscope, the molecular level, things are already happening. But as far as 2002 goes, the primitive streak signifies, according to this Presidential Council, the beginnings of a unique potential human being. So I mentioned the line that's drawn in the sand, the primitive node. This is where your finger goes in, and this is going to form more towards the caudal end of the embryonic disc. Now, some people call this the primitive knot or Henson's node. We're just going to stick with primitive node. Henson did a lot, in developmental biology. So, there's just a lot of things named after him. But the primitive node is this location where we're going to begin this process. And the depression within the primitive node is going to be the primitive pit. And so think of the elevated sides of this point, this focal point being the node, and the depressed middle as being the primitive pit. Now extend that further along as a trench, so we have the primitive groove, and this is going to be the middle of that primitive streak with those raised sides, and the primitive pit and the primitive groove are going to be continuous one with another. So once again, here we have an illustration showing our primitive node. You can see that it is more rounded, primitive pit in the center. Here we have our primitive streak, which is the entire structure, and the primitive groove is lying right down the midline. Here we have a transverse section, again at the level of our primitive streak, showing our epiblast cells. Now these are the cells that remain on the surface that do not migrate down to become endoderm or migrate down to become mesoderm. The epiblast cells that remain are going to be called our embryonic ectoderm. I just don't like the fact that we have that term here already, but we're going to get to that signification in a minute. But you can see these migrating cells coming down. Now, these migrating cells that are coming through the primitive groove here, these are going to be pluripotential stem cells, and we refer to them as mesenchymal. Now, they are migrating, and mesenchymal cells do have the capacity to migrate. So the first cells that come through, and you can see we have a cell here that's replacing our yellow hypoblast. These are going to become our embryonic endodermal cells. And then the cells pouring through that stay in the middle, those are going to be our mesoblastic cells. Now, that is really an oversimplification of this whole concept. But if you can understand all three come from the epiblast. The hypoblast cells are just placeholders and they go away. You're going to be in good shape right now. So we mentioned the first cells through are going to give rise to the endoderm. And again, we can see this in this illustration. And these other cells that are migrating deeply are going to become the mesoderm that stay in the middle. The hypoblast cells, they simply undergo apoptosis and are replaced by the endoderm. Here we can see a little bit later stage through an embryo. I believe this is a chick embryo. We're going to have our ectodermal cells on the surface, and we're already beginning to form our neural tube here. So this is a little bit later, but we have ectodermal on the surface, this flattened layer of endoderm, and then all of this stuff in the middle. Those are going to be our mesodermal mesenchymal cells. What I'd like to do before we move on to talk about our embryonic axes, let's watch just a short clip that sort of puts a little bit of this into motion and animation about the formation of the primitive streak. Here we are in the third week of development, and as we look at the epiblast, to the right we're going to see this is posterior marginal zone, and there is the formation of the primitive node. Now, as the embryonic disc grows, it's going to pull the primitive node forward, but it leaves this trail, this primitive streak behind. And differential growth of the embryonic disc versus the primitive streak is what will cause this structure to regress toward the end of this stage. As we saw in the video, the primitive streak really is the beginning of defining the embryo's axis. It begins in the posterior marginal zone, and it's going to extend and really define the anterior posterior axis. This axis is going to be reminiscent of where our vertebral column is going to go. Now, migrating epiblast cells are going to enter at the dorsal surface and migrate in a ventral direction, and so now we're setting up dorsal versus ventral, which is really epiblast versus hypoblast. And we're also going to distinguish right and left portions of the embryo through various molecular events that will occur. So cells that enter, we already saw that the first cells become endodermal cells, and then the middle cells become the mesoderm. Well, mesoderm, as we're going to see throughout development, can be divided up into several different types of mesoderm. And so the first cells that are coming through that are going to be mesodermal are termed chorda mesoderm, because these mesodermal cells are going to be moving anteriorly, and they are going to be moving in toward the region where we're going to form the head, and they're going to give rise to the notochord, hence the term chorda mesoderm. After those first mesodermal precursors come through, then the next mesodermal precursors are going to be lateral, and so those are going to be the lateral portions of the mesoderm. And so we've already distinguished different patterns of these migrating mesenchymal cells. And so as we begin to do this, we start to look at the molecular clues that are causing this to happen. And what we're going to see is that the anterior and posterior mesoderm migration occurs because of fibroblast growth factor 8. That is the FGF8 that you see. That's going to be expressed by the cells of the epiblast and the primitive streak cells. And it's going to be the expression of FGF8 that basically is the signal to go somewhere else. And so the primitive streak cells expressing FGF8 signal the migrating mesenchymal cells that are going to form mesoderm to move toward the chorda mesoderm or move laterally. Now, FGF4, these are also going to be expressed in the anterior mesodermal cells, and the FGF4 expression is going to signify that those mesenchymal cells are going to become chorda mesoderm. And so the FGF4 is in some way recruiting other mesodermal cells into the area to condense and give rise to the notochord. So, just in review, FGF8 from the primitive streak causes the mesodermal cells to begin to form, these migrating mesenchymal cells, to leave the epiblast layer, migrate between the epiblast and the hypoblast. Then FGF4 expressing mesenchymal cells are going to move toward the anterior portion and recruit other cells into the anterior to form chorda mesoderm. So even though when you look at a scanning electron micrograph, mesenchymal cells look the same at the molecular level, They're already beginning to distinguish themselves one from another. So in this illustration, we can again see that we've got our primitive streak in blue. And when we look in cross-section, we can see our migrating mesenchymal cells coming through. Again, they're leaving the place where we have FGF8 expressed. And then anterior to the primitive node, this is where we're going to form our notochord. And so our chordomesodermal cells, they're going to be coming in and migrating in an anterior direction, and that's going to be enhanced and facilitated by the expression of fibroblast growth factor 4. Now, in the video, we really didn't get to see the regression of the primitive streak. And when we talk about migrating and structures moving during development, I hope that you appreciate that it's not actually growing legs, using dynein and kinesin and picking up their suitcase and going somewhere else. What we're talking about is the relative movement of one structure versus another. And in this case, the regression of the primitive streak really is due to the fact that the rest of the embryo is growing so fast, this primitive streak doesn't grow. And so you're talking about a microscopic primitive streak that's outgrown by a 9-month-old baby. Hopefully you can get an appreciation for that. So because of this relative aspect, the primitive streak regresses after it forms those germ layers, after it signifies the posterior margin of the embryo, the anterior-posterior axis, then it's going to regress and completely go away. If it does not regress, then we can have some problems with tumor formation, which we will talk about in portions of our PBL sessions. Here we can see a relative map. Now, this is 10.5 hours where we're showing the formation of the notochord, we're showing the formation of the primitive streak. Understand this is relative. The notochord's growing so much faster than the primitive streak. But you can see this apparent regression of the primitive streak, which basically is caused by the outgrowth of the notochord in the developing embryo from this primitive streak tissue that is no longer required. Now, we've talked about the formation of the endoderm. We've talked about the formation of the mesoderm. Now, the epiblast cells that remain on the surface that are adjacent to the amnion, we now call them ectoderm. It's not that they've really changed or differentiated, done anything differently. They're just there. And this epiboly is a term. Epiboly literally means over the ball. And this is more in reference to how things develop in chickens, because they're going to completely invest the yolk sac. And in human development, we're only going to see a portion of this yolk sac type endoderm being invested and become part of our developing gastrointestinal tract. So this epiboly developing over the ball, over the yolk sac, we've seen so far the ability to designate the posterior marginal zone. We've developed anterior posterior axis, dorsal ventral. Now we're going to look at distinguishing what's going on with right and left. So, posterior marginal zone. How does the primitive streak know to begin in this location versus the opposite end? Well, when we look at the molecular events, we're going to see that in the posterior marginal zone, there is a transcription factor expressed. It's called VEG1. And it's going to be expressed, and it's going to induce the cells of the primitive streak to form. And it's going to inhibit the lateral cells from becoming primitive streak. So VEG1 is a very early inductive molecular event that happens to form the primitive streak in the posterior marginal zone. Now, again, we can go earlier and earlier. Well, what causes the VEG1 and what causes this and that and other? Well, we haven't quite got there yet, but we're beginning to put these pieces together in these pathways. We're also going to see that there is another growth factor family. This is called nodal, is the actual name of this gene. It's a member of the transforming growth factor beta, TGF beta family. And we're going to see that VEG1 induces cells of the streak. Nodal is going to become expressed in the streak cells. And then the streak cells are going to express a blocker of nodal. It's an antagonist, and it's called lefty. And so VEG1 is expressed in streak cells. Nodal is expressed in streak cells. Lefty is going to be expressed in the lateral cells, and that's going to block the formation of the lateral cells. So nodal is going to cause lefty to be expressed in the lateral cells, and that's going to block their formation. So now we've got the VEG1 and nodal being expressed, distinguishing themselves from the cells that are lateral, and this is all happening in this posterior marginal zone. Now, we're also going to see that hypoblast cells, they're also going to secrete a nodal antagonist. It's called Cerberus, because they're adjacent to the primitive streak cells, so we don't need to induce them either. So when we look at a cross-section through, we've got the epiblast on the surface, we've got the hypoblast underneath. So there at the surface, we can see VEG1 being expressed. We can see Cerberus being expressed by our hypoblastic cells that are not going to become primitive streak. But then again, we see nodal being expressed. So VEG1 nodal signifies primitive streak, and you can see the green on the right-hand side. And then we've also got our hypoblast cells expressing Cerberus, but at the same time, we've got our migrating cells coming in. We can see here in 2, these are going to be our migrating mesenchymal cells that are coming in and replacing the hypoblastic cells, and they're moving away, and we're starting to form our endodermal cells at this point. Now, right-left patterning. We know a little bit more about the pathways for right-left patterning here, but nodal still appears to be playing a role here, not just along with the factors that lead to posterior marginal primitive streak, but nodal is going to function in axis right and left patterning. So we can see that nodal is going to give rise to a certain pattern. However, the antagonist to nodal is called activin. So activin can actually block and stop nodal expression. And activin is going to lead eventually through a series of events to right-handedness. Whereas nodal and the nodal pathway is going to come via sonic hedgehog. This is a potent transcription factor. So sonic hedgehog is going to lead to nodal expression, and these are like dominoes falling. And nodal expression leads to the formation of PITX2. PITX2 appears to be the factor that will signify these structures are going to be left-handed structures. They're structures on the left side of the primitive streak in the axis. So as we look at cells of our early embryonic disc, here we can see our embryonic disc. Here's primitive node, primitive streak. We're beginning to form our notochord in this location. If we're going to be right-handed structures, then activin is going to signal. It's going to signal down through bone morphogenetic protein. We're going to have a fibroblast growth factor family of signaling that occurs. We also have another bone morphogenetic protein that assists in inhibiting nodal. There's our friend that we've been talking about before. But then FGF8 is going to turn on a transcription factor that inhibits the expression of PITX2. So if you inhibit the expression of PITX2 through this activin pathway, the structures are going to be right-handed. However, on the left side, we have the expression of an early transcription factor, sonic hedgehog. Sonic hedgehog is going to lead to the expression of a factor that inhibits bone morphogenetic protein. You can see how these are antagonistic of each other. On the right, BMP inhibits nodal. In this case, BMP is turned off by these earlier signals, allowing nodal to be expressed, which then signifies the expression of PITX2, and it leads to left-handed structures. So we understand more of the pathway of left and right, more so than we do the exact spot of formation in the posterior marginal zone, the primitive streak. But you can see at the molecular level, we can't visualize any differences, but at the molecular level, we have already been specified for right-handed versus left-handed structures. So fibroblast growth factors, they're critical in playing a role. And as we look at this huge family of growth factors, we see they specify mesoderm. They are going to cause the mesoderm to migrate. They are going to halt mesoderm formation, and they're going to induce the ectoderm to prepare to form a neural tube. We're going to see that that's going to be a critical event that leads to number 3, the initiation of neurulation and the formation of our neural plate that's going to give rise to our central nervous system. So fibroblast growth factors, they assist in our pattern formation, the formation of our mesodermal germ layer, and much more. Now, our notochord, this is going to be derived from our chorda mesodermal cells. That's going to be the precursor to our vertebral column. It's going to be the site where we're going to see our future vertebral bodies, or the vertebrae of our spine. And so the notochord defines us as a group, the chordates. And as we look at notochord formation, recall we're going to start with that chorda mesoderm. And that chorda mesoderm, as it uses FGF4 to recruit other mesodermal cells, we're going to form a solid notochordal process that is going to extend from the primitive node and extend forward as a solid cellular cord. Now, this is going to grow forward between the ectoderm and the endoderm in our mesodermal layer. And it's going to grow forward until it reaches the prechordal plate. Now, you may have seen this in some of our earlier animations, but the prechordal plate is an area where the ectoderm and the endoderm are fused together. No mesoderm can get between. And this is going to be the future formation. This is going to be the location of our mouth, or our stomadeum, in the anterior end of the embryo. So here we can see our illustration. We're looking down from the surface in the amnion. Here we have our primitive pit. The notochordal process is extending under our ectoderm and between our ectoderm and endoderm, and it's going to extend as far as the prechordal plate. Here's the prechordal plate, the future site of our mouth and stomadeum. Now, if we cut a section directly through the midline and look at this cut, what we're going to see, this is anterior prechordal plate. Here we have primitive node, primitive pit. Here's our solid notochordal process extending forward. Now, that is going to remain, and it's going to continue to extend, but you see how the primitive pit is at the very beginning there of our notochordal process. This is going to extend forward within the center of our notochordal process and begin to hollow out the center of that notochordal process and create the notochordal canal, so that we basically have a hollowed notochordal process that is continuing to extend forward. So in this illustration, we can see we have continued to extend our notochordal canal forward. We've gone as far as the prechordal plate. That's as far as we're going to go. And that notochordal canal is continuous then with the primitive pit and with the amniotic fluid that exists within the amnion. Now, the prechordal plate, this is going to be the location of our future stomadeum and mouth. And this is going to be attached ectoderm to endoderm, and we will call this the oropharyngeal membrane. Our gastrointestinal and respiratory tract, it isn't open continuously to the environment, and so this is going to eventually form the oropharyngeal membrane. No mesoderm is going to be between this. Now, at the opposite end of the embryo, as we've seen in our illustrations, the cloacal membrane, the future side of the anus, is going to be the same. It's going to have our endoderm mesoderm attachment at the posterior end, and this is going to form our cloacal membrane as we get towards the posterior side of the embryonic disc. Now, as the notochord continues to form, it's going to be a major inductive factor in the embryo, and it's going to be the notochord that is going to induce the overlying ectoderm to undergo neurulation and formation of our central nervous system. So the notochord, not only is it structural, but it's going to induce the major aspect of our central nervous system to begin to form in the appropriate location. So it's a primary inductive tissue in the early embryo. So as we've mentioned, the notochord is going to induce the neural plate. And all we're going to see of the notochord, because really in the human body, the notochord is an embryonic structure, an inductive structure, and then it goes away. So the notochord only is going to remain as the nucleus pulposus of each intervertebral disc. The rest of the notochord is going to completely degenerate and be overridden by all of our other tissues. Now, our notochord, and we can see this illustrated here, our notochord is a hydrostatic organ. It's going to be filled with water. And where we left our notochord, it had a notochordal canal that was fluid filled with the amnion. But what's going to happen is the floor of the notochord and the underlying endoderm are going to undergo apoptosis. And so you can see in this illustration, all we have remaining is going to be a notochordal plate. That notochordal plate is going to be just the roof of our notochordal canal. And here you can see that now we have a continuity between our amnion and our umbilical vesicle, the yolk sac. And what was the primitive pit we now refer to as the neuroenteric canal. In cross section, you can see how we've got the roof, our notochordal plate, and how it's connected and adjacent to our endoderm, but there's no floor to this. Well, this is not going to remain that way, because we are going to begin to roll that notochordal plate back up. The cells of the endoderm are really going to drive this rolling up as they grow and push back toward the midline with this connection point. And as that happens, we're going to roll this back up. We're going to seal off the ectoderm beneath, and we're going to reform a solid notochord that has a middle hydrostatic channel within the notochord. Now, why we didn't just leave it the way it was, I don't know. But we're going to go through that stage with the neuroenteric canal, reform and roll back up that notochordal plate into a hydrostatic, fluid-centered notochord. So, these are the processes that we've gone through to form our 3 germ layers: ectoderm, mesoderm, endoderm. We have formed our notochord. And in doing so, we have laid the foundation then to form all of our germ layers and all of the structures that come from them. So in this illustration, we see our trilaminar embryonic disc, which comes from the epiblast. That's going to form our intra-embryonic endoderm. It's going to give rise to much of our lymphoid tissue. It's going to give rise to some of our endocrine organs. We give rise to accessory digestive glands, the epithelium of our GI tract, portions of our respiratory tract. Here we have the ectoderm that remains. Ectoderm, certainly our epidermis and skin, enamel of the teeth, lens of the eye. But we're also going to produce the neuroectoderm. We haven't gotten to this yet. And this neuroectoderm is going to give rise to our central nervous system cells, retina. But we're also going to see that our peripheral nervous system, cranial and sensory ganglion nerves, the medulla of our suprarenal gland, pigment cells, these are all going to come from a subset of these neuroectoderm called neural crest. Everything else is going to be mesodermally derived. So go ahead and begin to think of these groups of tissues. This is going to help you as we go through the remainder of the course to know what portions of which organ and what tissue is derived from which particular germ layer. That's going to be important for you to commit to memory. Now, last, just want to mention this little structure. It's not something that gets a whole lot of attention or has a ton of significance, but it's the allantois. The allantois is a small diverticulum, which is a blind-ended pocket, and it's off the yolk sac. The allantois, you can see here, extending up into the connecting stalk. This is going to give rise to portions of our umbilical cord. And it's going to be this allantois that is important for the formation of some blood and the development of portions of our bladder. Now, in the birds, in reptiles, it has a much greater function and significance in development. But this will contribute to portions of our urinary system as we go through development. So since we can see that in this illustration, I wanted to make sure that we pointed that out. Now let's return and finish off our video of gastrulation. Here we can see continuing to grow. This is the ectodermal growth. You can see the primitive streak. Now we're beginning to induce the overlying ectoderm to form what is our neural plate in the beginnings of our central nervous system. We're going to be getting to that in the next podcast. But this finishes off the development and the view of the ectodermal growth that happens during this third week of development and the processes that happen during gastrulation. Now this is a very simplistic view of mesodermal growth, but they're going to look at both the surface view as well as what's happening between. And first we're going to see primitive streak cells giving rise to our endoderm. And these epiblast cells migrate in, and I hate they left them the same color, but they're going to migrate in and they are going to replace the hypoblastic cells. So the first cells moving in are going to become our definitive embryonic endodermal cells. And then the next cells that migrate and take up that place between the endoderm and ectoderm, we're going to color those red. Those are going to be our mesodermal cells. And remember, we have lateral mesoderm, quarter-mesoderm. In this particular illustration, they show the mesoderm just like a layer of paint being poured underneath the ectoderm and on top of our yellow endodermal layer. So there comes our red mesoderm filling all of our spaces up to the precordial plate and extending back to the cloacal membrane. Now in this case, the formation of our notochord, recall it starts at the primitive node. It's going to extend forward from the primitive node toward the precordial plate, first as the notochordal process with a notochordal canal that will follow. We will hollow that out into the notochordal plate that will then round itself up as the endoderm completes itself, forming our definitive and final notochord.